Hello, everybody. My name is Joe Ryan. I'm one of the faculty here at the Whitney Lab and would like to welcome you to tonight's event, and thank you very much for coming. Remind you that next month we have Dr. Sheila Patak from Duke University talking about extreme movement. It should be very interesting. Tonight we have with us all the way from Ottawa, Canada, Professor Kenneth Storey, who is the professor of biochemistry at Carleton University in Ottawa. Just a little background on, he's also the Canada Research Chair in Molecular Physiology. And a little background on Professor Storey. He did his PhD at British Columbia and then went on to be an assistant professor at Duke University. And let's see, he's won many awards, including he's a AAAS fellow and a member of the Royal Society of Canada. He has over 800 publications, and he works on some fascinating animals that can do amazing things, and he's going to tell us about those right now.
Yay! I'm mic'd, I hope. You should never clap before a performance, because then the terrible things that could happen later on. I was also promised high school students here I could torment, and unless your high school students have significantly white hair, I have a feeling it didn't show up. This is all about a phenomena that happens in nature except to you. You can't do this, and you're one of the only creatures that can't. It's about metabolic rate. You are warm in your body because you're breaking down food, because you're biosynthesizing new molecules, because you're using and producing energy in the form of ATP. I work on animals that start out alive. They start out warm, they start out wet, they start out as snails, they start out as giant squid. Then, for a long period of time, they get dead. You can't tell if they're not dead. Some of them are actually frozen solidly, and you can clink them in your glass. But then, the good news, right at the end, they come back to life. So what I study is turning off an entire organism, off. Then how you survive when you're off, and then on. Now, the reason I do that is it's fascinating. I get to go around the world and have other people pay for it. I think it's extremely interesting, but other people who know my work go, this isn't so much interesting as useful. So here's the thing. Young people tend to do really dangerous things. They snowboard, they skydive, they do things where at the bottom of a hill there's an ambulance. Well, sometimes their organs become available prematurely, and they don't tend to pass away where I can get them and put them into me. So this is all about old people, rich people who need organs because of the 1970s. I won't go into detail. Then other people say, you know, there's a serious problem. We have injuries where arms and legs are removed. We would like to drop the metabolic rate of those organs and then put them back on the person that's still alive. And this is a specialty of your Department of Defense. So we work with people that mean well. We work with people that want to do good, and I do my thing. So we're going to talk about how you have a metabolic rate. We're going to talk about how other animals drop their metabolic rate. We're going to talk about the tools that they use, and then how I can use the tools amazingly. There'll be giant squid at the bottom of the ocean. There'll be bears in space. There'll be primates that are hibernating, and most of you primates. This is an animal that perfectly exhibits what's going on. There are thousands of species of rodents and squirrels that actually go underneath the ground in the winter and hibernate. They start out at 37 degrees. They start out running around. They gain weight. They get fat. They go under the ground and they stop their metabolism. They go into metabolic arrest. They live in suspended animation. These guys are 5 degrees because they picked a very cold hole in Canada. Their brain is at 5 degrees. Their heart is at 5 degrees. Their body is at 5 degrees. They're cryopreserving all of their organs. And then what happens when you wake them up is, boom, boom, they actually come back. You pat a squirrel that's hibernating, it makes itself warm. It shivers and it turns on heat and it comes back to life. You can't do either of those things, and they're sort of amazing. This is another mammal. It's a bat. Bats hide in caves. The caves are cold as well, and the bats drop their metabolic rate. They hang there all winter. In the spring, they're so strong and so good, they just unhook and fly out. But bats aren't ground squirrels. Bats and ground squirrels are very different mammals. They haven't had a common ancestor for millions of years. So here's the thing. Bats do exactly the same thing as ground squirrels do. They look the same, they act the same, their metabolism is the same, and all the biochemistry I'm going to teach you tonight is exactly the same. There's one way to turn off a mammal, and it's the way I'm going to tell you. It also turns off a mammal heart. When it's taken out of a mammal, it turns off a mammal kidney. And even more amazing is, it's not just mammals. Turtles drop their metabolic rate to 0, and they do the same thing. Toads do it. Everybody does it. You just are stuck with a high activity lifestyle. You have a high Vmax, you have high oxygen needs. And I'm never going to be able to hibernate a whole person, but I'm only after your tissues. I'm only after your organs. In case you don't believe that given enough money and enough graduate students, I can't take out your heart and make it live outside you, this is a primate. Come on, you're all come on, show me your opposable thumbs. You know it. This is a primate that hibernates. This primate hibernates for a short period of time in torpor bouts. Some of them hibernate for weeks at a time. The primate does exactly the same thing the ground squirrel does, and it's going to do exactly the same thing everybody else does. The truth is that all the tools I'm going to tell you, you have. You have all the enzymes, you have all the DNA, you have all the genes, you have all of the mitochondrial metabolism, you have everything, you just can't get it together. These animals turn off 40,000 things at once. Little switches in the house. Click, 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 click. What you do is you would like to do it, but you've lost the organizational ability. You've lost the molecules in your brain. You've lost the blood proteins you need. So as a whole organism, you're pretty hopeless. Now, remember, I have a bad view of humans because I'm going to take out your organs, so it all fits. This is a snail. This is a snail that lives in deserts everywhere. This is a snail that if you've eaten escargot, you've eaten. When you take away water from this snail, it immediately sucks itself back into its shell and stops breathing and stops metabolism. When I take these snails and find them in Morocco and Italy, I take their body temperature, they're hotter than you. They're hotter than your body temperature. They're nearly 100 degrees Fahrenheit. They're 40 degrees centigrade. And their metabolism is 0. Now, here's the thing. You thought with hibernators that, oh, they're cold. Of course they're slow. They're cold. No, they're not. They're slow, and then they get cold. These are slow, and then they get hot. There's every chance I can take your organs out, do my little metabolic tricks, and leave them at room temperature. I don't need to preserve them. I don't need to cool them. I can just keep them at room temperature until they need to be transplanted. This is also important for the Department of Defense, who know full well that you can't cool down battlefield injury people in the battlefields they're on. This is a toad. If you don't believe a snail has anything to offer you, this is a real vertebrate. For 11 and a half months every year, this toad is underneath the ground, so dry that he's crispy. Sometimes it doesn't rain for 10 years. So for 10 years, they're under the ground, dry and crispy. They're not only dead, they don't even look good. But in Arizona, the rains come. Boom, 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 boom. Huge amounts of water comes down, and suddenly there's a million toads. Toads estivate at high temperatures. Vertebrates dry down. Add water, stir, you got toads. You got millions of toads. This is a really good model for hot animals that he can preserve his organs. His brain is fine, his heart is fine, his kidneys are fine. This is a frog that you may have heard of. It's Xenopus. It's the clawed frog. It comes in Africa. In Africa, entire countries dry up. You've seen the things about migration of wildebeests, right? That because it's the dry season. Millions of square miles will dry out. These frogs suck it up, literally, dig into the mud, and get dry and crispy until it rains again in Africa. They're great model systems, and we believe that when I show you what they do, you'll know that it's true that anyone can do it. They do exactly the same things as a lemur does. These are animals that can survive without oxygen. I don't want to go into it too much, but we're all subject as humans to heart attacks and strokes. If I cut off your oxygen, you have about 2 minutes of brain life. And if a stroke cuts off oxygen to part of your brain, that guy's not coming back. Your heart has maybe 5 minutes of health left, but you're going to die. This is a vertebrate with a brain and a heart as complex as yours that survives without oxygen for 9 full months. I could envision a time when I could take out your heart and brain, drop your metabolism to 0, keep that heart and lung fine like a turtle, boom, in for transplant. These animals drop their metabolic rate by the same mechanisms everyone else does, and there's tons of invertebrates. There'll be a test at the end of this about animals you know and care about on the beach as you drive home. So you'll know about animals on the beach. You'll never be able to look at shrimp scampi again. You're talking about an extreme animal. You're talking about an animal that went to the wall here. You're talking about this is a wet, hoppy frog. There's hundreds of millions of them. They exist all across the top of North America. When it gets cold, and trust me, in my town it's minus 15 degrees, they freeze solidly. That's not a bit of snow on a frog. That's a hard, crunchy frog. If you tap it, it sounds like metal. It's all frozen. But you know what? If after weeks and months of this you put it in your hand and warm it up, you open your hand and it's alive. It's alive. It hops away. So, I don't care if you think about estivation or drying down or hibernation. It's not cold. It's turning off your metabolism and then finding yourself in a bad environment. You never see, in Canada, huge groups of frogs hopping past your town migrating. You're a frog, it's going to be minus 20. You better freeze or get a good undertaker. So, this is an example of mammalian hibernation, because you're a mammal, and all animals are sort of the same. The first thing they do is turn off all of their metabolism. Boom. They go into a suspended state. You couldn't tell that animal was alive if I gave you a year. The low body temperatures that come are because they pick somewhere cold, perfectly natural. I do not have to give these animals drugs. The only drug I can give to humans that will drop your metabolic rate even a little, severe amounts of opium. These animals drop their metabolic rate to 1%. You can drop your metabolic rate by 1%. They're 99 times better than you. The reason is your environment suddenly sucks. It used to be great. There used to be milk. There used to be honey. There used to be good things on television. And then suddenly, you got no food. You got no hope. You better turn off your metabolism and wait for better times. I could insert a Trump joke here. I understand that's not good. Told not to do that. Although that's especially true in Texas, because they're a carry state. I'm telling you, I got to move back and forth in Texas. So, this is an example. This is what scientists do. So some of my slides are what scientists do in the lab. So we're funded by taxpayers' dollars. I get a lot of your tax money. I feel terrible about that. No, I don't. We go into work, and we do what you do. We have coffee, we talk to our friends, we flip through the sites, see how our shows went. But this is what we do then. We take an animal and we measure its metabolic rate. How much is it breathing? How much oxygen is it using? How much CO2 is it making? How warm is it? How fast is energy turnover? And what you find with these animals is their metabolic rate falls off the chart. See that? Whoosh, whoosh, whoosh. You can't do that. You can't turn off all of your metabolic rate because this is turning off 40,000 different processes. After you turn off your metabolic rate, it depends where you are. If you're in the cold, you cool down like a brick. These guys are dead. Essentially, they cool down like a brick. If you're small, you cool down like a very small brick. If you're bigger, you cool down like a very big brick. And if you're huge, it takes you a long time to cool down. And if you're a bear, you don't cool down. You're just too big. You're too big and you're too fat and you've got too much fur. They don't cool down. But they turn off their metabolic rate. Everybody does this, and you can't. That's what I study. That's why I study it. So what I'm going to tell you, all animals do. They turn down their metabolic rate, then they freeze. They turn down their metabolic rate, and they have no oxygen. They turn down their metabolic rate. Ooh, I can use this again. So here's what happens. Here's the sort of core science-y things here. Trust me, I've got a lot of papers. If you type in Ken Storey lab into Google, you find my lab. You find hundreds of papers and review articles. There's pictures of our animals. It's 2-year-olds love my site. Metabolic rate reduction, I'll tell you how that happens. It happens by 2 mechanisms. The first thing is you've got a set of enzymes, every animal does, that reach out and turn off the rest of the cell. So there's an enzyme you can turn on. It reaches into the cell and turns off hundreds of processes. Then most of your genes turn off. Right now you're actually turning on and off your genes, just sitting there, not thinking very much. You're turning on 22,000 genes. This costs money and this costs money, and you got to stop doing it. So metabolic rate regulation is about stopping the expensive things you're doing, and then they turn on a very few genes. The genes they turn on allow them to live in suspended animation, and these are tremendously important genes. These are genes that we have funding for, and we're looking to see how, once you have cryopreserved a heart, once you have slowed down a heart, how do you keep it in that state? This is what your cells are doing now. Every single cell in your body is doing this. You're turning on and off genes, and you're making message RNAs. It's expensive. Costs you ATP every time you put 2 nucleotides together. These cost you thousands of molecules of energy. You got to stop doing that. Then, once you've made messages, you put them onto big factories and you make proteins. Every time you put 2 amino acids together to make a protein, every time it costs you 4 ATPs. 50 to 60% of the energy in your cell is doing that, even when you're not running around, and you got to shut it all off. If I reached into your bodies now with drugs and turned these 2 things off, you would die. You need these. My animals have learned to live without them. You're pumping ions. You're in with the good ions, out with the bad ions, in with the good ions, out with the bad ions. They stop that. They stop their cells leaking. They stop their cells pumping. They save energy. They turn off these enzymes by reaching out with protein kinases. You're breaking down carbohydrates, fats, proteins. You're biosynthesizing new carbohydrates and fats. You got to stop that. 70 to 80% of your energy is running through the mitochondria. The reason you're 37 degrees, the reason you're hot and sweaty, is your mitochondria are sucking in fuel, making ATP, and causing you to live the good life. You got to stop that. They turn off their mitochondria. So you turn off everything. This is what you have to turn off. And I'll talk about each area, and then you can look at individual papers if you're excited. So this is the other thing we do when we have students. When we have students, we take years of their lives to work at selfless tasks about measuring everything. And then they get jobs at good universities, and then they take more of your tax dollars. It's a sad cycle for you, but it's working out for me. I'm a big star. Now, what they do is, you may have taken science in school, and what they say is, well, you've got to have a hypothesis. You got to have a hypothesis. You figure out what's going on, and then you test that hypothesis, and then you look at your data and see if your hypothesis was right or wrong. That was when we had unicorns and elves. My lab is orcs. Orcs all the way down. So, you got 22,000 messages. I don't have time to figure out which ones are important and which ones aren't important, and I'm not smart enough to figure it out. So what I do is I measure them all. We have developed a word called omics. There's mRNA omics, and what that means is I measure them all. There's a word called proteomics. What do? Come on, all together now. I measure them all. There's phosphoproteomics, and there's microRNAomics. Oh, it's so exciting. I measure all of those too. Those are really cool. I measure, are you using fuels? And the answer is always no. I measure which fuels have you turned off, because what I want to do is take a human heart and turn off those fuels. What I want to do is take a human heart and figure out what the ground squirrel is doing and stop it. Oxygen consumed. You need oxygen. And if you don't believe me, I'll go to this guy. He's right at the end. And I will test how much he needs oxygen with these. Who can test what oxygen he needs and has 2 thumbs? This guy. I put my 2 thumbs, one on each of his carotids. You need oxygen. You can't choose not to use oxygen. These animals do. They say, I used to use oxygen, I'm now turning myself off, and I won't use oxygen anymore. Talk about impossible. You guys are junkies. Just breathing in and breathing out. So, the first thing that happens in all cells of all animals is your protein kinases turn on. They come up from a deep sleep. They listen to the blood and they listen to the brain with molecules and say, oh, we don't have any food, we don't have any water, it's getting cold, we better shut down. There's 700 to 1,000 of these. Some turn on and some don't turn on. And the trick is being able to figure out which ones are which. Once they turn on, they turn off making messages. You've got to stop that. They turn off making proteins. They turn off pumping. They turn off fuels. And they so turn off your mitochondria. They just turn off your mitochondria like a light. It was like facing the early Mike Tyson. So there's a few of these protein kinases, they turn on. What we do is we measure which ones turn on, and then we gather them in a pile. Because if I can take these kinases and turn them on in a turtle, or in a snail, then I can turn them on in your discombobulated organs, so I can take your organs. The second thing that happens is you've got to turn off all of your genes. You've got to stop making messages and stop making your genes be active because they're so expensive. As it turns out, half of the audience here, half of you, have one organ that does that. Even though you're old, it still does it. I'll give you a hint. And it's only half, so it's not you. You have one organ that knows this trick. It can turn off all of the genes. So it takes a perfectly good cell, it squashes down the nucleus. It ties up the DNA. It makes it so the DNA can't be active unless it's sent out of the body searching for somewhere to be. Yeah. Now, you guys aren't all old enough for me to tell you what it is, but it'll be on the quiz afterwards. And then a few genes are activated, and these few genes allow you to live as a essentially dead animal in suspended animation. The mechanism that all of my animals use in all of their cells, the brain cells of ground squirrels, become sperm. They shut off all of their nuclei, they don't do any more thinking or activity, and then they wait and wait and wait until hibernation ends. It's called epigenetics. The reason you've heard of epigenetics is this is a transgenerational phenomena. What your parents ate made marks on their DNA. They passed on that DNA in eggs and sperm, and now you have that mark. What your grandparents were frightened by, your parents were influenced by, and then you caught as a piece of DNA. Schizophrenia. passes through generations as an epigenetic marker. When you look at schizophrenics who've taken their own life versus schizophrenics that haven't taken their own life, they have different markers, but they both have the schizophrenic markers. Epigenetics is tremendously important. You'll be hearing about it. It's why you have to have individual personalized medicines. Medicines work for some of you and then not others. Some medicines work. It's because of these marks on your DNA. What I do is found that these marks also occur in my animals, not across generations, but during turning off their metabolism. The marks happen here, and in the beginning nobody believed. The marks happen here, nobody believed, but it's how you turn off all of your nuclei. So, there's mechanisms, and I'll tell you all of them. So, this is like the most sciencey part of this. There's 3 main mechanisms. You mark DNA, and you mark it in a way so it turns off. You mark proteins that bind to DNA, and you mark them so they turn off. And you make tiny, tiny, tiny little RNAs called microRNAs. You're going to hear about these too. They are at the forefront of all modern disease medicine. So, this is what my animals do. They do it in order to drop their metabolic rate. So, the trick here is you got to turn off your cells. And when you look around the body, it's unusual for you, but cool for others. DNA methylation. You mark DNA with a big methyl group. These are big hydrophobic groups. So you take them and you stick them onto DNA with an enzyme. Stick, 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 stick. Then, when you try to turn on the DNA, you can't reach it. Transcription factors can't get in. DNA can't unfold correctly. DNA is blocked by these big, fatty molecules. And you know what happens when you warm up? Do you know what happens when you wake up? Do you know what happens when you come back to life with water? Demethylation. You just take the methyls off. Secondly, you modify histones. These are proteins, and I'll tell you what they do and why they're exciting. They've been known since the 1950s, but their function was only found out recently. What their function is to wrap up DNA in a tight little bundle so it turns off. So if you have histones and they're wrapping up DNA, you're off. But if you free the DNA from histones, they're free. Then the DNA turns back on again. And the third one is microRNAs. So we'll talk about these 3 mechanisms. And this is what we do in the lab. This is what costs money. This is why we spend long hours. And this is why it's semi-exciting. You have DNA without methyl groups on it, and they're turning on and off genes all the time. You're good. There's an enzyme that will add methyl groups. It's one enzyme. We know it. There's 14 different kinds of it, but we know it. And what happens is methyl groups hit the DNA and you're done. You're done like dinner. Until another enzyme reverses that. Interestingly enough, this enzyme runs on SAM. You've heard of SAM. It's called S-adenosylmethionine. You can buy it in health stores because it's supposed to make you healthy. It does not. This is what S-adenosylmethionine is actually for, methylating your DNA. So, if I ground all of you up, but not in a bad way, I do it after I took out your heart. So you'd already be dead. I'm not cruel. If I ground you up, husbands and wives would have different methyl markings. 90-year-olds would have different methyl markings than 30-year-olds. And even identical twins who've lived different lives, one smokes, one doesn't smoke, different methyls. You have different marks on your DNA. Modern DNA technology is based on sequences, but I can tell every one of 7 billion people by looking at all of their methyls. My ground squirrels, my animals methyl and shut down, and then this gets better. Histones. Histones are proteins in the nucleus. Their job is to hug DNA. Here's DNA. When DNA is free, you have an open chromatin. You get messages, it's open and active. When histones change and hug DNA, they condense down. So when you look at DNA that's open, it's actually big and fluffy. When you look at DNA that's closed, it's actually hard and crunchy. So a hibernating ground squirrel has different DNA, hard and crunchy. Oh, active ground squirrels, oh, they're all happy. You modify the histones, and this changes how they act. So we study histones. We measure them, we analyze them, we look at different histones in different animals, and we try to modify their nucleus with histones in order to turn them off. So we can do this with real animals, in invertebrates mainly. Also, in your nucleus are different categories of proteins and enzymes that we didn't know about before, and there's 3 categories of them. You have writers, a series of enzymes, you have erasers, another series of enzymes, and you have readers, another series of enzymes. So science is funny. Anything that you don't know about is forbidden. Then you find it, and nobody knows what it does, and then you find out what it does. You do it in that order, and it sometimes takes decades. These enzymes were known, but nobody knows what they do. Now we know they crunch DNA. They crunch DNA. They keep it from making new messages. So you have significant methods and enzymes to turn off your DNA, phosphorylations to turn off your proteins and metabolism. You have these. You got this. You can't put it together. You can't turn off your own DNA. You've forgotten it as a human. Oh, if you were a lemur, you'd remember. So the trick here is transcription and translation. Those things at the top of the slide are very, very expensive. You methylate DNA, you modify histones, and microRNA is yet to come. Let me show you papers, but you don't have to read the papers. You just have to see sort of what we do in the lab on a day-to-day basis. This is a paper where you get together with other groups and you say, hey, a warm, wet ground squirrel, a cold, hibernated ground squirrel, do they actually methylate their DNA? And you do expensive experiments, you do difficult things. Science has to do things over and over again. You're not just able to do them once and walk away. And the correct answer here is yes. They methylate their DNA, they turn it off, and this ground squirrel's brain simply shuts down. It's going to turn on again by the opposite enzymes, readers, writers, erasers. Secondly, histones. This is a paper about histones. So your answer is, hey, you told me that DNA crunches up. Come on, prove it. You have to prove it. We measure all of the enzymes. We measure all of the histones and how they're modified. We look at the DNA and see if it's crunchy. Boom. Drop the mic. Yes. So we get money for proving things. And here's another thing about ground squirrel brains. When a ground squirrel goes into hibernation, it builds up tau proteins, T-A-U, tau proteins, because they're not degraded because it's so cold. You are right now building up tau proteins. So there's tau proteins in the brains of ground squirrels and in our brains. And the older you get, the more you get. Tau proteins then are fixed in your cells by something called phosphorylation. Remember those protein kinases? Man, they're having a bad day here, they phosphorylate tau. So in your neurons are huge plaques of tau, great ropes of these proteins. And as we get more and more of them, we get neurological diseases. We used to be sensible. Men would die in war, women would die in childbirth. You got married when you were 16 and really looked good, and now you're in your 40s. Of course we're going to die of this. When a ground squirrel wakes up, it melts its tau plaques. Whoosh. Not so much us. They get worse and worse, and then we don't remember our pudding. So, you have to look forward to us working on the brains of ground squirrels. We maybe solve this Alzheimer's thing. Or, you know what would be the worst thing? I do all the work, I publish all the papers, and then just as I'm about to tell you, I forget. Because I have... Man, that'd be terrible. MicroRNAs. So I'm going to tell you about them, then we're going to talk about them. They are awesome. First, microRNAs didn't exist. My lab discovered microRNAs, and they told me they didn't exist, and then they existed. They are weird and wonderful. They're tiny. They're just tiny. They arise like mosquitoes in a cell. There's 1,000 of them. So as soon as you turn off a cell, you make 1,000 of these little things, and they begin to appear. They appear in the nucleus and in the cytoplasm and in the mitochondria and in the endoplasmic reticulum and near the membranes. And we didn't know what they did. We now know what they do. They're highly conserved. Your microRNAs are the same as all other mammals. They affect gene activity and they turn it off. These are molecules that my animals, hey, that's awesome, dude. MicroRNAs, turn off, just what I'm looking for. And they make microRNAs. The cool thing is every disease now, every disease has a microRNA fingerprint. We can suck your blood, measure the microRNAs, and tell you about tumors you have. We can tell you about where you're having problems in your different organs because microRNAs hang out in something called exosomes, which we isolate. This is not technology yet that's going to your doctor's office, but okay, you know health science. We're all going to die. If there was high school students here, we go, hey, high school students, this is going to be awesome for you. No medical breakthroughs are for us. Come on, you know that. They don't even make movies we like anymore. Gah! My animals make microRNAs. Subsets of 1,000 different ones. They turn them on and they use them to turn off everything in the cell. So turning off is the theme today. MicroRNAs are brilliant. Here's why microRNAs are good. I can feed microRNAs to animals and they go into the cells and turn them off. I can take organs like your heart and dip them in microRNA. They suck up the microRNA and turn your heart off. I can also get microRNA into your cells in other ways. The hard thing about histones and DNA is that it's a long process. The cool thing about microRNAs is I can imagine a time when a nice young person slides their motorcycle under a bus. I grab out their hearts, their kidneys, their lungs, and their livers, put them in a soup of microRNA. I got them turned off. Let me tell you about microRNAs and how we found them, because we had to develop new computer programs and new methods, and then what they do. If you're interested, on the website, there's review articles that tell you about turning things off. Before, nobody worked on microRNA. Now, everybody works on microRNA. So, what you have to do first is show there is microRNA, and you have to show that the microRNAs change and control things. So the first thing you get to do is you get to measure them. So that's really cool because they're brand new and nobody's measured them before, and it gives you this thrill of discovery. So what we discovered is some microRNAs out of 1,000 actually increase. And when a microRNA goes up, they turn things off. And they don't just turn one thing off, they turn a whole thing off. MicroRNAs increase and you don't make any more muscle cells. You just stop. As soon as the microRNAs go away, you start to make muscle cells again. You turn off your cell cycle. You turn off your low oxygen response. You don't make any more red blood cells. There's diseases that have red blood cell proliferation and white blood cell proliferation in them. We know the microRNAs that can shut that down. It's a brand new frontier, and it doesn't just work for hibernators. So we measured them all in hibernators. That was the previous slide. This is a marsupial. Oh, they're adorable. Smaller than kangaroos, but marsupials. Marsupials are not placentals. Marsupials and placentals are totally different. They come from a different place, but they do exactly the same thing. They lower their metabolic rate. They go into hibernation, profound hibernation, and they change microRNAs. So not only does every mammal we know, including a lemur, do this, but so do marsupials. I'll show you slides. So do turtles, so do snails, so do frozen frogs. It's a thing. We know what tissues they arise in. We know how those tissues shut themselves off. And I'll tell you a little bit about how you can look for organ preservation and look at other diseases. This is a lemur. This lemur is in Madagascar, but what happens is they take them around the world and breed them. And so there's a colony in Paris, France. It's hard work. I got to fly to Paris first class on their dollar. I've got to go see the Louvre again and again that someone else pays for. I have to eat pâté at nearly every meal. And these breed like lemurs, honest to God. And they've got too many lemurs. So the French government wanted them to cut the colony back. We took some lemurs that were awake and some lemurs that were hibernating who were going to be culled anyway, and we just took out their tissues. So no endangered lemurs were harmed during the course of this experiment. In Canada, I also point out that the lemurs were French, and we don't like the French, so... There are 6 papers in a row. You can read them and you can understand them, because I'll show you what they do. Why would a primate have to hibernate? They live in a bad neighborhood. You know the lemurs you've seen, right? Those big, ugly lemurs with the striped tail. They're not only ugly and mean, they're bullies. They push the little lemurs out to the edge, and the problem with the edge in the tropics, you know, the wind blows for half the year. It's wet. It's wonderful. They can eat. They're dancing, singing on their cell phones. When the winds shift, it's dry. No leaves, no food, no nothing. So these are mammals that can't get away. They can't fight off their enemies. They can't get a plane ticket. Hey, come on. I know 10% of you are Canadians. Don't tell the rest. Don't use the word about or house. And for heaven's sakes, don't say a. Because they have to face a terrible time and they can't get away, they drop their metabolism. But here's the cool thing. They're not cold. It's never cold in Madagascar. It's warm and dry and warm and wet. This is a primate whose organs I can take out and preserve. And you're a primate, roughly at room temperature, whose organs I can take out and preserve. Later tonight in the parking lot, I've been in New Jersey. Stuff happens. Things happen. I need tissues? I got a guy. They drop their metabolism profoundly, but they don't drop their temperature very much because, duh, it's warm. Their metabolic rate, remember that metabolic rate? That's good. Boom. Boom. This is an animal shut off. This is a primate out of it. You publish papers, you measure everything, and what you find, they do exactly the same thing as a frog, exactly the same thing as a toad, exactly the same thing as a ground squirrel. They have signaling in their tissues. Their tissues strengthen themselves and fight off free radicals. They turn on new cool genes just like all the rest. They change their expression of genes, and they change their metabolism. They reorganize their insulin pathway, and their guts are protected. There's a thing where we need more transplantable human guts because of irritable bowel syndrome and because of Crohn's disease. There's an active call for how do we turn off guts and transplant them into people that increasingly need it. So the gut was important here. So, do they use microRNA? Hell yes. They not only use microRNA, but they make new microRNAs that we'd never seen before. So we measured all of their microRNAs, and that's what happens. Look, some of them go up, and some of the new ones go up. So this can help you. If I can know the microRNAs that turn on this animal, I can put those microRNAs into a human tissue, and then I can depress the metabolism of that tissue, and that's what the Department of Defense wants to do with arms. That's weird. It's a weird world. You don't have to use low temperature. You can use regular temperature. So right now I've described a guy with big grants and hundreds of students who measures things. We measure things. Red means down, green means up. We have thousands of papers. Well, we have 800 papers. And what does it mean to you? How does this help you? And the correct answer is microRNAs. Once you know them, you can add them to a tissue, an organ, and turn that puppy off. Hey, we could actually turn puppies off, but no one wants to do that. We know the microRNAs that change in a liver. So if you had a liver to give, we could take out that liver, drop its metabolic rate, and then have it available for transplant for long periods of time. Younger women do have a liver to give. It's very strange. If a young mother has a child with liver problems, they go into the mother, take about a third of the liver, replumb it, and put it in the kid. The kid has a liver and it's fine. The mother then just regrows that liver. It's a thing. What about muscle atrophy? Did you go to the gym this morning at our age? It didn't do you a blind bit of good. Walking back to your car, your muscles forgot you exercised. You may as well have a latte. But did you know that ground squirrels lay down and don't move for 6 or 7 months of the year? And they have no muscle atrophy. They don't decrease their muscles. Your muscles went to hell during the NFL season. You know when you sat there, didn't move, and had Cheetos? Bats fly away after 8 months, strong enough to fly, and they haven't moved in any of those 8 months. So what we know is what microRNAs change in order to keep your muscles healthy and happy, even though you're not exercising. I have a theory that if you lift something heavy enough to make you go, ugh, you really shouldn't be lifting that. And I've got back problems like Mike Greenberg has to prove it. So, we do heart transplants with UCLA. We take bits of human hearts, we look at transplantable organs, we try to turn them off and keep them in metabolic suppression. We have heart and stroke grants and diabetes grants from Canada because they're interested in, if you have a stroke victim, Ken, could you sneak up on them and put some microRNA in them and make them happier for longer? We do laser surgeries in Illinois where you take an organ, treat it with a laser in order to drop its metabolic rate, and then microRNA and limb preservation with the Department of Defense. It's important work. I hope it works. I have no way of proving it, but it's been a fun ride. So you often wonder, what do bears have to do with going into space? You've wondered that. The high school students have wondered that, and I blame marijuana, but that's not the point. When we go into space, terrible things happen. Our bones melt. Our metabolism screws up just and does bad things around mTOR and AKT, and our muscles atrophy. So if you've seen the movies, there's two kinds of movies. There's one kind of movie where Lorne Greene, who takes thousands of ships to another planet, where you live and you breed and you go to school and you have babies, and then 50 generations later, they canceled that show. It's called Battlestar Galactica, and Lorne Greene is a Canadian. Or you've seen the movies that are more realistic, where they put people into pods and keep them in pods for 15 years, and then when you get there, there's, yeah, and then they come back to life. What we say is we have hibernators that have massive amounts of problems. There's deaths of you sitting bed rest, and they happen in microgravity, and they happen when you're old. And we have hibernators that have solved those two twin problems. So we ask, is it going to be like this? And every once in a while, it's going to be like this. Keir Dullea used to be in movies like this. Or is it going to be you exercising for 900 years with your offspring on the way to another planet? The only way it can be like this, got to be like a bear. You just got to be like a bear. We send some scientists, so I'm on a project with the European Space Agency, we send graduate students from France up to Sweden to do bears. It's dangerous work, they're grad students. So our question was simple. If I have a hibernator, and after 9 months of lying around doing nothing and getting fat, they're tremendously fat, how do they do it? Do they exercise in their sleep? Are they magical animals where we can learn about their genes and their microRNA? Or are humans doing something that I can reverse when I measure microRNAs? And the correct answer is this one. So you study animals that hibernate. Oh, there's them again. Oh, there's them again. There's them again. And this is with bears. So what you do is you take a bear in hibernation. They do not hibernate well, and they only have one gene left. Kill, kill, kill. So you got to anesthetize them. You anesthetize a bear in hibernation, and you anesthetize a bear active, and you work its muscle. You work its muscle. This is just a stimulator. You've had that when you've had a bad knee, right? You've had that stimulator at the top and the bottom of your knee. That's what we do. When we do it with humans, look at this. This is bad, and this is doubly bad. If you've had even a little bit of bed rest, I don't mean staying in on Sunday till 10:30 and reading the New York Times, but if you've had a lot of bed rest, your muscles get worse. It would take you a year to make this better because we don't train up very well. If you take a bear after months and months and months, look at that. There's not even a statistical difference here. They do not get rid of their muscle. They have ways of keeping their muscles strong, and it has to do with microRNA. So we have pieces of bears in our lab, we measure microRNA, and we say, look, here's how you can change a muscle and make it strong. In detail, this is a bit of a detail, when you exercise a bear, what happens is there's a decrease in overall synthesis. So as we get older, our muscles do actually get softer. We are synthesizing less protein, and there's also a bad degradation thing as well. Bears overcome that. Bears do less degradation, more synthesis. And so what we do is we study their genes. Remember how we measured all their genes? So we measure their genes, we look and see which messages are up, we look and see what histones are modified. So we're good to go. We have this under control, and we may get rid of bed rest problems and send you to Mars, your children to Mars. Okay, your great-grandchildren to Mars. Bears are in hibernation. Bears don't move. Here's a thing for younger women. Female bears have babies while they're hibernating. They don't move. Nothing happens. Do you remember the screaming at all with the birthing? Do you remember saying things about your husband? Do you remember saying you would never have another baby, and then you found yourself pregnant again and angry at your husband again? They go to sleep in hibernation, and they wake up with a baby, and they don't even know it. Bad news, it's always a baby bear. They are literally asleep in hibernation, thinking about exercising, and their muscles exercise. You gotta like that. So there's things that seem to you like magic. There's things that people are working on that's not common sense. But once you get to the molecules, once you know the molecules, once you can run the data, you can find amazing things all around you in nature. This is an amazing animal. We thought this was the key to organ transplants. The National Institutes of Health thought for 12 years it was the key to organ transplants. I apologize for you not being able to buy a second home. This is a frozen frog, and we thought we could freeze organs. Frozen frogs are amazing, but it's turning out to be way hard. They change their microRNA in an organized fashion, and that microRNA drops their metabolism, but we've never been able to take a human organ and tissue, freeze it solid with ice in it, and get any purchase at all. However, we can make food safer. We can take foods, we can drop their metabolic rates, we can turn them off, then we can freeze them, and they're happier and healthier for longer. We can take embryos, we can take eggs, we can take sperm, we can microRNA them up, and they last longer. They're viable for a longer period of time, not human reproductive organs, but there's a lot of animals that you want to breed, but their eggs and sperm are very hard to breed. This is a freeze-tolerant turtle, and when you dig in, there are so many changes that it's hard to deal with them all. So the real problem with freezing wasn't that it didn't work. Freezing works, and it wasn't that microRNA didn't help. It's that we don't understand what's going on. And so that happens. Sometimes scientists do things, and then you're at a break. Hey, we don't know what's going on. We stop. Somebody's going to help us. What freezing animals do, though, and you can read about this, it's sort of cool, is freezing animals like the frogs make new genes. They turn on genes that you don't have. They're so new that we call them numbers and names. These genes, when I express them in human cells, allow human cells to freeze. So if I freeze your cells, they die. You can't take ice at all. You're terrible. When I put this frog protein into your cells, they survive freezing. Freeze-thawing, they're amazing. So the good news is the freezing story has given us magic proteins that we now use as cancer drugs. So if you have a question, it's complicated to say, but it's turned out really, really interesting from frozen animals. So, you live by an ocean. Why am I talking about all these crazy animals? Marine invertebrates drop their metabolic rate all the time. Marine invertebrates drop the metabolic rate with high oxygen and low oxygen, and high temperature and low temperature, and high CO2 and low CO2. Oh dear, that's climate change, sorry. With high pH and low pH, they shut themselves down, and some marine invertebrates are awesome. This is a sea cucumber. Have you ever tasted a sea cucumber? They taste like death on a beach. They're worth $4 billion a year. In China and Asia, they eat these. And the problem with these animals is the ocean is getting hotter. Shh, don't tell anyone. And these animals shut themselves down. Instead of going with the flow, they shut themselves down, and you can't make them into food. So they were interested in us sequencing their genome, and they said, hey, Ken, why are they shutting off? Why are they turning themselves off at 40 degrees? And the correct answer was microRNA. These animals are fine at normal temperatures. We're good, we're getting fatter, a little warmer, we're moving faster, and then suddenly at a magical temperature, they're done. These are the microRNAs that increase. Once these microRNAs increase, they turn off metabolism, and this guy's in a state of suspended animation. It will protect them from the worst of temperature change until cold water comes, but it's hell on food services, so they're interested in us turning on these animals. So that's a marine invertebrate that's really important that uses microRNA. You've asked yourself, right? What's my giant squid doing tonight? You have. In the day, huge squid like these, which are called red devil squid, they're killer squid, they'll bite you, are at the bottom of the ocean. The bottom of the ocean. It's very cold, it's very dark, there's tremendous amounts of pressure, and their metabolism is shut down. They're sitting and waiting. Do you know what they're doing? Trying to avoid sperm whales. So what happens is sperm whales dive down and eat these guys, but they're in a state of suspended animation at the bottom of the ocean. Every night, something called the deep scattering layer rises. They come up and up and up and up and up thousands of meters. They get to the surface of the ocean and they're going so fast. They eat, they kill each other, they get eaten by sharks. If you're on a boat hunting for giant squid and you're doing experiments, don't fall off the boat. So, what are these animals doing? At the bottom of the ocean, they're metabolically depressed. Their microRNAs are high, their histones are grinding on their DNA, they've turned everything off. And as they come up, temperature, pressure, they get more and more and more active, 100% active. These things can go the length of the boat faster than you could drive a motorcycle. So it works for giant squid at the bottom of the ocean. It works for sea cucumbers in China. So here comes the quiz. Here's some marine invertebrates. You've either seen them, or you've hit them with a hammer at your table and eaten them. You've seen them under a rock. Of these marine invertebrates, which ones drop their metabolic rate and increase microRNA? Which ones? Look at them, look at them, look at them. All of them! Yes, they do. It's a thing. It's a principle. Hey, if ground squirrels do it, if lemurs do it, come on. They all do it. Every different kind of marine invertebrate does the same thing. High, low oxygen, yep, turn it down. High, low temperature, turn it down. High, low salinity, turn it down. It's a thing. Here's another slide. And these are different marine invertebrates, and there's even more of them. What about these guys? What would you guess? Which one of these can turn on the metabolism? All of them! Oh, you're doing well. You didn't do well on the first question about sperm, but after that you got over your nervousness. You're running an A, but not an A+. What about all the things that you put onto rice beds here and eat? Everything that's crunchy. What about crabs? What about shrimp? What about how many of these drop their metabolic rate and live in suspended animation, both at the bottom of the ocean where they live, as well as in the inshore when they're stressed? All of them, all crustaceans do, all. What about these guys? Come on, they're octopi. Yes, mollusks drop their metabolic rate. Octopuses go into profound metabolic depression. Cephalopods are types of mollusks. All mollusks go into metabolic depression. We study mainly mollusks, so this is cool. And at the Whitney lab, several people study mollusks. The most complicated and fast, the most sessile and weird. This is a nautilus, an ancient animal. And the ones you can eat. That's the snail. All mollusks do it. Even funny animals that nobody studies. Now, the reason I can study these is all I have to do is grind them up and measure microRNA. These are crabs that we use for other types of studies. These are crabs that live on seashores, and we check them for global warming, for high and low temperatures. These are weird crabs that you've never seen before. Every time we check, all of them do it. So what I've told you is actually true because there's hundreds and hundreds of papers. And what I told you is also real because I think it's going to come to fruition. I think there's going to be more and more of this. We are not done, despite my advanced age. Scientists can go along as long as they have money. I don't even have to walk well. And we have new directions and new exciting students that are really interested in this. We're going to look at metabolic rate depression. We're going to look at those protein kinases. We study whether or not organs get stronger, like in a bear, or weaker. MicroRNA is only in its infancy. We measure microRNA always. There's thousands of new numbers in my email now. Epigenetics, turning off. Lifestyle, cell cycle. I think it's important. I wish I was 29 instead of 109. But we're not wasting your money. We're working hard. I have to visit marine labs all the time and drink coffee. So, my students do well. Everything is going on well in the world of science. I think it's practical and not just esoteric. The Canadian government thinks it's good. And bless your hearts, a number of agencies in the United States also have given me a slight vote of confidence. So, do you have questions? Let me try it. The opposite of protein kinases, I've just turned on the lights. Okay, questions.